talk is a dialogue across disciplines, and our aim today is to find some common ground on which we can explore continuities across forms of liquid life and connections between the well-being of life forms beyond the human. The arts and the sciences traditionally occupied much common ground, not least in the types of historical research expeditions that are referenced in the work of Alberto Baralla, who is one of the artists participating in live streams. Today, however, our research institutions are arguably less architecture of universities and the way they work administratively. And it's going to kill my video because it might be easier. As we confront an increased climatic instability, as well as our...
Sorry, Lisa,
do you want me to continue, Lisa?
Yeah, maybe.
Okay. So yeah, so the okay. So I'll pick up disciplinary boundaries can be felt in degree courses and in the architecture of universities, but as we confront an increasing, sorry, I thought I was muted. But as we confront an increasing environmental and climatic instability, as well as our current epidemiological crisis, it is more urgent than ever to find ways to open the doors that separate forms of knowledge. Interdisciplinary conversations are often slow conversations, exchanges that evolve in the translation of the processes of knowing that we take for granted, assimilated into our cognitive and communicative forms. Interdisciplinary conversations can often be awkward too, but this is not a bad thing. In starting from what we don't know, posing questions that might or might not be relevant to our interlocutor, we cross bridges into other worlds. Contact with other knowledge, other life forms, can at best develop in us a deeper sense of tact. This is what the feminist philosopher Karen Barad proposes in her article on touching, the inhuman, therefore I am. She writes, theorizing, a form of experimenting, is about being touched. What keeps theories alive and lively is being responsible and responsive to the world's patternings and murmurings. Doing theory requires being open to the world's aliveness, allowing oneself to be lured by curiosity, surprise, and wonder. Theories are not merely metaphysical pronouncements of the world from some presumed position of exteriority. Theories are living and breathing reconfigurings of the world. The world theorizes as well as experiments with itself. That's the end of quote. If we reach out and establish contact, we are acknowledging and activating our porosity, recognizing that the membranes that seem to separate us from the world, our pores, are in fact full of holes that open and close in response to myriad others. Today, we're happy to welcome as our guest interlocutor, Professor Terry McGenetti, Professor of Environmental Microbiology here at the University of Essex. Terry's research, it seems to my totally non-scientific mind, this is Lisa speaking, by the way, through me, to be all about survival, about reckoning with how we understand the conditions for life to exist, persist, and ideally prosper. In his work on microbial ecology and diversity, he seeks to understand how microbial communities function and interact to influence major biogeochemical processes. He explores the microbial ecology of ancient salt deposits, investigating haloarchaea, residual forms of life that exist trapped inside salt crystals, to understand how they manage to survive over geological time. and how some species have adapted to flourish in less extreme salinities. He also researches carbon cycling, exploring the biodegradation of biogenic volatile organic compounds, which have a big impact on the climate. In asking how microbes cope with and flourish in hypersaline and polyextreme environments, his work moves speculatively beyond our terrestrial frontiers to consider conditions for life on other planets. We have been in informal conversations with Terry for around a year now, through a common interest in how art and science can work together. In today's iteration of this ongoing conversation, myself, Diego, and Lisa have each developed reflections and questions we would like to explore with Terry. So we will move across and look back to interconnected topics. Throughout the talk, please feel free to use the chat to send comments or to post questions. Let's keep this sort of open and informal. We can pick these up as we go along and at the end too. So also, I guess, feel free to interrupt.
Yeah, speaking of interrupting, Lisa, let me know which visual material you want me to share when as well. I think.
Okay, perfect. Should I try my connection and see?
Yeah,
is the audio okay?
The audio is okay right now. Yeah, we can try. If not, we can just Emilio can take over again.
Yeah, I mean, I have the text, but I don't have the at hand the images. So
I've got them. Just let me know when you'd like me to share and what you'd like me to share, Lisa.
Okay, I'll let you know. Thanks. Okay, so we've divided the talk into three. Spend a little time thinking about questions we really wanted to pose.
No, I don't think it's going to work. I think we're just going to have to.
Yeah. So are you familiar with the work, Diego? Also, I guess. Let me see. Okay.
Sorry, we can just start off with the questions. But yeah, I think what Lisa was about to say is that we've divided the session into three different sections. So Lisa's going to be talking in the first one, talking about tides and seas in the bodies. Then Emilio's going to take over. So Lisa's meant to be doing this first section, and then Emilio's going to take over and ask some questions himself. And then I'm going to finish off by talking specifically about the exhibition and the artworks and just seeing, yeah, I guess trying, sorry?
Bueno, acá. Listo. No, nunca me avisaron. Pensé que se habían olvidado de mí.
Okay, someone, yeah, microphone.
Yeah, someone needs to mute. I'm trying to mute, but it's not letting me.
Yeah, I don't know who. Okay.
Okay, perfect. Yeah, and then I'm going to finish off by just getting Terry's, Terry's perspective on the exhibition and everything.
So I can start on behalf of Lisa. I mean, I'm going to read. I have the text here, and so perhaps you can share the screen, Diego, with the images, and I think we can all make sense of it. I mean, I know the work, so.
Yeah, so it says where to share the images, so I'll share them when they come on.
Okay. Human bodies contain seas and rivers. Water flows through our human bodies in myriad ways, and many of the works in this exhibition invite us to become attuned to these through our own sensory feedback loops. As 2/3 of water, divided into 97.5 saline and 2.5 freshwater fluids, our bodies mirror exactly the Earth's liquid composition. These similarities remind us that all life emerged from water and continues to depend on it. Just as rivers are the veins of the earth that transport water and nutrients, so do humans' arteries channel vital fluids that irrigate and enliven our bodies. Most of our bodies are permeated by saline fluids that constantly circulate through us, through our entire connective structure of the fascial system that links bones to cells in gelatinous forms, sorry. We're also home to a tiny reserve of freshwater rivers and lakes that makes up our cerebral spinal fluid, a vital liquid that pulsates through our heads, removing toxins. From your perspective, Terry, what can be gained from considering this mirroring of the saline and fresh waters that run through the entirety of our bodies? What common variables and factors affect the well-being of humans and rivers? How do rivers self-regulate to return to balance and health?
Thank you very much, Emilio, Diego, and Lisa, first of all, for inviting me along today. I hope you can hear me okay, and I hope the internet sort of hangs in there. And for taking me outside of my comfort zone as well. So I'll try and answer that as best I can. I'll pick up a couple of things. First of all, the toxicity issue and the relationship between rivers and our body, and how they self-regulate. And if we first of all think about the human body and what's going on there, it's basically a series of human-driven pumps, reservoirs, and membranes that take toxins, anything from food additives to drugs, many toxins that we inhale. It takes them to the liver, where the enzymes, the catalysts, can break them down, and then they can be transported to the kidneys and excreted. So it's always been considered a very human-driven aspect, detoxification. But there's a growing element, a growing understanding that actually there's a lot going on in the gastrointestinal tract, in the guts, and a lot of the detoxification and degradation is carried out by the microbes in there. And I'll probably talk about that a bit more later, but that mirrors very much what's going on in our rivers. I was brought up in a chemical town in the northwest called Widnes, that's on the banks of the River Mersey. And like many rivers, in the 1970s and 80s, it was essentially a corpse. And I'm very pleased to say that many, many years later, more than 40 years later, it's revitalized. It's very much a diverse, flourishing river. And the killing of the river was due to the import of pollutants and such like, and the recovery was partly due to some human intervention, dredging, and so on, but primarily due to, yes, the flushing of the water, but also the actions of the microbes. The hidden heroes that can detoxify collectively a huge array of organic compounds and degrade them into innocuous products. And therein lies the parallels between the human body and the restoration of rivers.
Amazing. And then following on from that, I'm just going to pin myself as well. Emilio, we can share the duties of these questions as well.
Okay.
Yeah. So again, we're reading from Lisa's questions. We want to talk a little bit about the research on salt crystals, where you collaborate also with emerging scholars. So as we prepared for this talk, you shared with Lisa, Emilio, and myself a really inspirational video by one of your PhD students called Maria Magliulo, which I'd like to bring into dialogue with an artwork by the Venezuelan performance artists Yeni and Nan, which I've shared with you previously. So I'm going to share with you the video and the artworks as well. Let me know if you can you see the video,
Oh,
And I think I need
Yes,
to.
I mean we're seeing the Vimeo page. Yeah.
Yeah.
So. Yeah.
Can you hear it as well? Can you tell me if you can hear it once? There's no sound. Yeah.
No, we can't.
you may need to enable that
Yeah.
the audio. If I can.
Yeah, from sharing.
Sorry, 1 second. Apologies about that. Yeah. I'm gonna stop sharing and then share screen. Sorry, I'm trying to get the actual audio. Do you share sound? There we go.
Okay.
No, Emilio, it's not letting me share audio on my screen, unfortunately. I'm gonna send it to you. Okay. Apologies for that, everyone.
I think I have it, but I haven't downloaded. I'm sorry.
I'm sending you the video on WhatsApp.
Okay.
Apologies for that. I shared video earlier today, and it's now asking me to install a video, install something. It's terrible.
No worries.
Okay,
We should have brought
fine.
Maria in to give a
I
live
know.
performance.
I know. Apologies.
No worries at all.
Okay.
Do you have it?
Yeah.
Nice. Perfect.
Can you see it?
Yeah. And then I can share the images as well after.
Like the Dead Sea.
Let me, sorry.
Today, I want to take you on a journey of many dimensions. We'll start from Hebersalem, like the Dead Sea, and its wide prairies where, as the sun shines, unusual flowers bloom. Salt crystals. I know it's not really a place that teems with life, but let me tell you, it's just a matter of scale. That's why I will need you to become as small as the tip of a human hair. And now that we are just a few hundreds of micrometers, an entire new world of tiny, salt-loving microbes opens to us. Let me show you around. There is Dunaniella salina, a green microalgae that is fixing the carbon from the air to biomolecules, giving off the oxygen we all breathe. There is Salinibacter ruber, a bacterium that throughout its evolution stole some techniques that we call genes to survive in these environments, and it stole them from proper extremophiles. Haloarchaea, single-celled organisms evolutionarily distinct from eukaryotes like us and bacteria. These Haloarchaea challenge the limit of life at high salt by accumulating salt in their cells. Not a common thing to do, normal cells would just shrink. Another challenge they win is against the intense light. Their red pigmentation acts as a protective buffer by absorbing and dissipating excess light energy. As the sun shines though, the environment around us changes. The water evaporates and we get trapped, together with our microbial fellows, into tiny pockets of brines encased within the salt crystals. There's no need to be scared. Haloarchaea can teach us how to survive. For example, they have taught me that the oxygen will be consumed soon in here, so we would need to learn how to breathe without it. They may be able to produce their own terminal electron acceptor to replace the oxygen. They have also taught me that there's no need to bring our own packed lunch. They are masters in recycling nutrients. And perhaps the most interesting thing that they have taught me is that even if there's nothing to eat, they have a strategy. Instead of producing new proteins, they use part of them as a carbon source, so food. Their metabolism slows down, but this adjustment allows them to challenge another limit to life, time. They are able to survive in this crystalline cage for hundreds, millions of years. After 3 years, I still look at the crystals I created in the lab in awe and wonder, imagining how crazy things really are like inside the crystal. Then, through this lens, I look up in the sky and I ask myself, is there life elsewhere in the universe? Mars geologically is not much different from Earth. Today it looks like a desert, but in the past it was warmer and wetter, and salt crystals are buried in the surface. And I wonder whether there are any Martian microbial fellows awaiting to be discovered.
Thanks, Emilio. And I'll share now my screen because it doesn't require audio.
Okay, so
Can you see the?
Yeah, yes. Wait. Yes, we can.
Okay, amazing. So it's these images of these performances.
Of Jenny and Nan.
Jenny and Nan, exactly.
Venezuelan performing artists.
Exactly.
So I'll read Lisa text. It's striking to me how both of these works, Maria's research and Jenny and Jan and Nan's performance in the saline, how do you say, saline or saline? Well, saline environments of the Caribbean coast invites us to think about expanded frameworks of life on Earth. Jenny and Nan worked in the early 1980s in Venezuela, but for me, their practice resonates strongly today. They engage intimately with the environment around them, here immersing themselves into a place that would appear hostile to our fluid bodies. Through introspection, the practice of centering attention deep within our bodily fluctuations, which is inherent to well-being practices such as yoga and meditation, they perform modes of slowing down and attuning to our and other forms of life. This, to me, has potential for how we consider the eco-ethical dimensions of art and of science. Engaging with salt, it seems, is a way for us to enter other rhythms where life unfolds beyond the human. At a time when anthropocentrism and human hubris is unquestionably linked to the economic and social paradigms that are extractivist and seek only to generate value from the Earth's resources, Maria's research asks us to consider the bigger story of our place on Earth. On that note, Terry, could you tell us more about the importance of salt crystals? What do they have to teach us about survival, and what about the geohistory, the history of the Earth before and perhaps after humans?
Great, thank you very much, Emilio, Diego. So thanks for sharing the video and the artwork there. So, and thank you to Maria, who did a sterling job. That presentation won the 3-minute thesis competitions. She's a second-year PhD student, and she's far more articulate than I am. So it was great that you could show that. I think we all know that salt's vital for health, and in fact, we're made up of about 0.4% salt, and historically, it's one of the most important commodities. It's right up there with water and oil, primarily because you could use it to preserve food, either directly, for example, with salted fish, or indirectly by fermentation processes. And that gives you more nutritious food that can last longer and be used over during lean periods like the winter. And as a consequence of that, empires are being built on salt, like the Venetian Empire. Wars are being fought over it, and people have been paid in salt. So it's a really important commodity. Less so now because of refrigeration, but historically, it was, and you could see in those images the red color of the saltans. And what you're looking at there is seawater that's been brought in, channeled into shallow ponds, and to make salt, to make salt as a commodity. And the red color is primarily due to Haloarchaea, these types of microbes that Maria mentioned. And they're red as a form of protection against the strong ultraviolet light, amongst other reasons. And they came to prominence actually in a bad way, in that when people were dealing with salted fish, occasionally you'd get a disease called a red heat, and the fish would turn bright red. And this was due to dissolution of the crystals and the halite, the Haloarchaea hatching out and eating the proteins and converting them converting the fish into a putrid red mess. So they weren't very popular at the time. But they're also, the haloarchaea that live inside the salt crystals are also important. For example, in fermentation, and my very first bit of work as a PhD student involved isolating, culturing one of these microbes from Thai fish sauce called Nam Plat, one of the many fish sauces that are used in Eastern Asia as a condiment, but more than that, as a major source of nutrients. So they have great value as well. And we should always remember that we're consuming haloarchaea as we're consuming sea salt. And the main focus of my work that you alluded to there was the fact that they don't just survive, haloarchaea don't just survive inside salt crystals for years, for a few years. We have evidence now that they can survive for tens of thousands, hundreds of thousands, and potentially millions of years. And the big question for me, and that Maria's PhD project actually, is to try and understand how they can do that. And that's kind of important in the sense that it does make us think about what is life and what is death. And it does raise the question of, could things live forever? But it's also important because most life is in the slow lane. It's living with very low energy levels and just ticking over, just doing enough to repair its biomolecules, its proteins, and its DNA. And that's where we're coming at with this project.
Thanks a lot, Terry. That's fascinating, and I think well, yeah, perhaps it's a coincidence or not that it's the specific same. It seems that it's the same type of archaea, no? The one that we're seeing in Jenny and Nan's work, and yeah, the crystals that we saw before. Yeah. Which is very interesting. And talking about coincidence, I mean, I received two papers on my email, like preparing for this talk, and one of them was just yesterday talking about archaea as well, sort of. It's this paper that suggests it's about the what I think it's an open discussion about how eukaryotes formed, which is complex cells that make up life. As I understand it, we are made of those kind of cells, and pretty much everything living, complex living. And so what it says there is that it's exploring the idea that this archaea protruded. I mean, it was a process in which I think Lynn Margulis said that it was archaea with some bacteria that came together and formed these complex cells. And this paper is exploring the idea that it wasn't necessarily a bacteria, but that the archaea produced the bacteria or something. This is very sort of my understanding. I'm sorry, I'm not.
No, it's a fascinating story, and so yeah, Lynn Margulis was an amazing scientist, and she brought to light the endosymbiotic theory, so which is, as you said, involves the engulfment or coexistence of a bacterium with another organism, and the internalized microbe ultimately became our mitochondria. The mitochondria of eukaryotes that is the powerhouse that generates the energy, and still retains a bacterial genome, and implants the chloroplast derived from so-called cyanobacteria. But what's kind of new, I mean, that's in the last 30 years, but what's new is this idea that eukaryotes, that is our cells, the fungi, certain protists, plants, all derived from within the archaea, just a specialized branch within the archaea. So that's the real new story that's emerging.
Yeah, I think it's fascinating, and also brings this other thing that I have a specific question. And so these archaea, either Margulies' way or these new findings brings us pretty close to the archaea that we're discussing. No, bring us human close to that sort of simple to the archaea, and also we have tons of evidence now that what we call human is made of various different things, right? Like bacteria in our stomach, and you just now you were saying that when we eat salt, we eat bacteria. So it's a very, very, very complex organism and exchange between different sort of fungi, many organisms, the microbiota. So for me, it's really interesting, not so much in explaining that this is actually something that's true, because I think there's a lot of evidence, but in the implications of that. And so it's very interesting. I mean, the first question would be, so how come we can, it's a complex one, but how come we can call ourselves human? And what do we mean by that?
Yeah, that's a massive philosophical question.
Yeah.
I think I'll probably answer it slightly pragmatically.
It's fine.
It is a really, really important question. Are we more non-human than human? And I guess the key word there is more, and what we mean by being human. But you know, in terms of numbers, being a scientist, we've got to have a few numbers thrown in there. Approximately half the cells in our body are microbial, which is always an astounding piece of information. And in fact, if you think about it, whereas human cells, each one has the same set of genes, many of these microbes are different from each other, have a different set of genes. So there's something like 100 times more different genes in our body that are microbial than human, which kind of tells us that there's great potential there for microbes to affect us. And of course, the microbes don't see us as human, they see us as an ecosystem in which to live, on our skin, in our armpits, in our guts, and so on. And we know, we've talked about what they can do, and that's really what's crucial. It's been known for a while that they can extract energy from food in the gut, they can provide growth factors like vitamins. I mentioned they can detoxify pollutants, and they can also protect us from disease by kind of crowding out the bad guys, the bad bacteria. So we've known that for a while. What's really new and exciting, and comes on to the question of how they might be affecting us is, you know, the things that they do. For example, some of them will produce chemicals, those living in our armpits, for example, that affect the way we smell. For better or worse, you know, that they can be an attractant. So that's one way, and that scent can be perceived differently by others. They can stimulate and educate our immune system. And, you know, we have this thing called the microbiota gut-brain axis, where the microbes in our gut, there's strong evidence now they can influence our behavior to certain degrees. And then there's the kind of hygiene hypothesis, where because we've not had exposure to as many microbes as our ancestors or people living in more rural areas, there's a decline in diversity and various things like due to excessive hygiene and birth and infant feeding practices and changes in our diet, less fiber and so on, use of antibiotics, this all affects our microbiome. And it can affect the diseases that we become susceptible to, especially things like chronic non-communicable diseases like asthma and obesity. So I haven't fully answered your question, but I hope it's given you a flavor of the different ways in which microbes make us who we are, and the exciting field that you know, how we can control that or potentially control that in different ways.
I'm gonna pass to other questions, but before I just wanna sort of make a comment on that. I think it's fascinating, and I think it touches on a very important question, and perhaps it's not pragmatical, but it's not as philosophical as the other one. It's about agency. So if we're recognizing that you know, this organism can control our mood or our scent, and we can understand things as reproduction from there. We're talking about a very sort of complex ways in which agency, you know, interacts, and I think that's very important. And I don't think there's much time at the end if we can pick that up again. But I do think there's a lot of to say in terms of agency and ethics, you know. But that's another thing that we can discuss. But I think we can go with Diego and to see if people have more questions. But thanks a lot, Terry.
Thank you very much, Emilio.
Hey, Terry. Thanks so much for that. That was really, really fascinating. And yeah, like I said, so I'm going to be talking a little bit more about works in the exhibition specifically. And I've got about like 4 or so questions that I've planned out, but I don't think we're going to have time for all of them. And if there's some questions that maybe you don't have much to say about, we can just skip to another one, etc. So feel free there. Yeah. So I think, like Lisa said, having these interdisciplinary conversations can be a little bit scary or awkward, and I'm definitely speaking from within my own field of expertise, but within the full knowledge as well that these boundaries, these disciplinary boundaries, are porous, and with a desire to want to learn more about your expertise. So just sort of grounding where I'm coming from before I start asking some questions. But yeah, I'm gonna just share my screen really quickly. And we won't need sound, so don't worry. But yeah, I'm gonna go on this is our exhibitions website, by the way, if you haven't seen it yet. And on the exhibitions website, you can see all the artworks that are on display at ArtExchange, as well as we have an online screening room for works that aren't available on ArtExchange, and a few other things as well. So if you haven't seen it, we encourage you to have a look. But yeah, before speaking about artwork specifically, I wanted to ask you a more general question about, I guess, transdisciplinarity and collaboration across disciplines. So my personal practice as a curator and I guess a early researcher, it's very much informed by people who have who are joined together by a refusal to accept sort of these oppositional dualisms and binary thinking that's quite characteristic of modern Western thought. So dualisms such as nature and culture being separate, fact and story being separate, reality versus constructivism being separate. So those kinds of oppositions, I'm very interested in people who reject that kind of thinking, or who want to think beyond that form of thought. And the sciences are often placed on the side of objectivity, of fact, of reality, and humanities are often placed on the opposite side of that, you know, of story, of myth, of culture. And I think a rejection of these binaries is also an acknowledgement that, firstly, all these fears, they co-produce And it's also an acknowledgement that there needs to be more dialogue between these disciplines that are often placed in opposition of each other and that are separated from each other, as Lisa said in the beginning of the talk. Entre Rios, the project that we're working on, that this exhibition is part of, is at its core a very transdisciplinary project. We've tried to bring the sciences in with the arts, with anthropology, with a lot of different disciplines. And we're hoping to highlight the need for interdisciplinary dialogue and to start having this dialogue as well. So I guess my question for you is whether you see similar conversations about this need being had within scientific circles. So within scientific circles, do you see similar conversations being had about the need to have more dialogue with the humanities, for instance?
Thank you for that, Diego. Yeah, absolutely, we do. Especially, I wouldn't say it's the case in all areas of science, but certainly in the area that I'm in environmental science, where we're trying to understand how ecosystems can benefit us, can benefit society. There's this horrible term, but perhaps necessary term of natural capital, putting a value on nature. And at one level, you can think of that as being, highly objective and breaking things down into numbers. I mean, it kind of is, and we need evidence to be able to somehow convince people that it's important to conserve nature. But part of that equation, more and more, is what nature does for people in terms of its beauty. We have things called areas of outstanding natural beauty. We've had those for a long time. That the main designations are scenic beauty, wildness, and landscape quality, and so on. So I think it's becoming more and more important in the projects that we have, in how we use nature to benefit society. And I'm thinking here of everything from capturing carbon dioxide from the atmosphere and fixing it into organic carbon, through to people getting out and getting exercise in a beautiful place and improving their mental well-being. And more projects and more grant calls are demanding that we talk to each other. So we've kind of been pushed in some sense to do this, but it's also great to do so and to learn from each other in the ways that you've described. So yeah, I see it as a very, very positive move, and a move back to somehow the old way of doing research, of breaking down these disciplinary boundaries.
Thank you. I guess staying on the topic of interdisciplinary boundaries, we don't have much time, unfortunately, so I might just only ask one question, and I'm kind of trying to decide which is the one I want to ask, because there are a lot I want to ask you. But I guess, okay, I'll ask this one about Giannietta Varsi. I don't know if you had a chance to look through her work. We held a session to open the exhibition, actually, with Giannietta. She's a Peruvian artist, and she does a lot of breathing exercises, basically. And we had a sort of breathing exercise workshop to start off the exhibition a couple of weeks ago. The thing with Giannietta's breathing exercises, I like to think of them as a form of making abstract scientific information a little bit more concrete or tangible or understandable through sensing. So her exercises, they draw on scientific principles like homeostasis and other bodily processes that attempt to maintain equilibrium, and that both human bodies and bodies of water enact and carry out. So for instance, as part of our research for Entre Rios, we had a long program of speakers and talks with the artists that participated in making research projects for our digital platform. We had a Colombian scientist, a biologist, I believe, called Luis Camacho, present his research on oxygen levels in the Bogota River in Colombia. And having this presentation shortly followed by breathing exercises by Genietas, where we were gasping for air and really trying to embody what it's like to embody a drowning river, essentially. You really felt like you were, embodying the river, and it provided a certain understanding for Luis's work that I wouldn't otherwise have had. So I guess what I wanted to ask are, what are your thoughts on more sort of somatic, bodily, performative ways of relaying scientific knowledge? And if that's too abstract a question, maybe more a more accessible or interesting question for you would be, how do you think humanities and the arts can help make scientific knowledge more widely understandable, or what are your ways of making complex information like this like a bit more widely understandable to the masses? Because I know your work, you sent us some research that you did with on salt that was extremely accessible, extremely understandable. It had like amazing drawings, amazing images. So yeah.
Well, yeah, I thank you for giving me the option there, Diego. To be honest, it's the one of the artworks that I didn't delve into. There were several that I did that I found absolutely fascinating. But yeah, I'll address your more broad question, and I'm involved in a program on microbial literacy. That's why I sort of bang on about microbes all the time, because they govern the earth. Basically, they rule the earth in many ways. And there's, if generally for better, sometimes for worse, as we're seeing, which is why we're not meeting in person. But generally for better in producing the oxygen that we breathe, for example, half of it comes from microbes. So yeah, I think that the arts has a massive role to play in making any complex subject within science or whatever available and accessible to the public, and also just for bringing especially youngsters into the field. It's amazingly important. And I've tried to, there's all sorts of ways we can do this. And one of them is just a very simple thing, is to get kids involved with something called agar art, where you can take a Petri dish, you can take a bacterium, and you can draw pictures on that and see how it evolves over time as the different colonies from different bacteria grow, all suitably controlled, of course, with safe microorganisms. But there's many different ways in which the arts of all different types can benefit scientific understanding and certainly bring youngsters into the fold.
Amazing. I think, okay, I have a ton of questions, but I think other people do as well. So I'm going to open up to other questions. If they don't start flowing in, then maybe I'll get to ask some more of mine.
There's also a question already on the chat. Eva.
Exactly.
Okay, so you want to start with that one?
Yeah. So we'll start with a question from Eva, who says, what is the importance of oxygen levels of the river and its life? So what is the importance of oxygen levels to the life of a river?
Yeah. Great question, and it's absolutely vital. It's a sign that that most of the organisms, the higher organisms within a river, require oxygen in order to breathe. Fish, for example, crustaceans, and so on, and a lot of the smaller organisms. So they need oxygen. So it's absolutely vital, and all sorts of malpractices can lead to a reduction in oxygen not least dumping lots of organic matter, sewage, for example, into a river. That leads to a surge of activity by microbes that gobble up the oxygen and deprive its availability to other organisms. So it's a vital component of any river. Yeah.
Conversely, I also think it's quite interesting to think of. We discussed a lot throughout our meetings about what is a living river and what is a dead river, what makes a river dead, and something that Lisa was speaking about not long ago that I found really interesting as well, that is that in things that you would think are completely dead, like salt and things like that, there's actually a lot of life. Like I remember the images you sent us of sort of mud that you got from near the banks of the river in Wivenhoe that looked like just gunk that nothing could live in, but actually they were completely full of life. Yeah, I think that's an interesting thing to think about. But I'm gonna open up.
I'm gonna make a really fast comment on that, Diego. Also, yeah, I think we were also amazed on how some very complex ecologies, not only oxygen, but the right complex ecologies of bacteria were identified with the health of the river. Also, like a heterogeneous sort of ecology with, of course, of the right bacteria would be a symptom of health, or yeah.
Yeah, you're absolutely right. I mean, and this isn't, of course, just microbes. It's what ecology students do. They to understand the health of a river. They're one of the first bits of field work they do is they sample from different regions near the sewage treatment works. You've got your chironomid midge larvae and nothing much else, and further down you've got your damselflies and such like, and a whole greater diversity of organisms. So yeah, it's and that's related to the amount of oxygen and the amount of pollutants in the river.
We have another question from Jess. She is asking if you could expand on how the corpse of a river in your hometown was cleaned by microbes and how long that process took. Were certain microbes chosen over others, or I don't know if there was human intervention there in terms of regulating what microbes were there to clean it or not. But.
Yeah, thanks, Jess. So what I would say was there was intervention. So there was a lot of horrible sediments that built up with. Sorry, Jess?
I think.
Okay, so yeah, some of it was human intervention by digging and dumping, but the vast majority was just letting the microbes that were there. No microbe was added. Just using the native microbes to do their work. And it was their innate capacity to collectively be able to degrade a wide range of pollutants, as well as the flushing over time. And of course, there was the major important thing is the reduction of input and new regulations and new behaviors. But there was very rarely when you're cleaning up an environment is it useful to add a microbe to do the job. Usually they are already there, and that's an important issue. It relates to the idea that everything is everywhere and the environment selects. Certainly from a microbial perspective, they get around, that they are everywhere, and if there's something good that they can use as a carbon and energy source and grow, then they will do so and they will flourish in that environment. And they can grow on all sorts of nasty man-made chemicals that they've never seen before because they evolve so quickly as well. Does that answer your question, Jess?
Thanks, that was really interesting. really interesting, Terry. I wish I spent more time talking about microbes on the football pitch line when we did the Sunday football together. Yeah. Thank you. I'd love to know more, but it's a fascinating area. Thank you.
Thank you.
We have a question from Jane. Jane has her hand up.
Thank you very much for a fascinating discussion, everybody. Terry, it's really, I'm sort of coming back to one of Diego's questions, which is really, I actually listen to a lot of science programs on the radio. There's some fantastic programs now, and that's actually quite a new thing. It's really been in the last 8 years, I think. And one of the things that really strikes me, which kind of goes against this dualism that Diego was talking about the arts as imaginative and the sciences as being about fact, is just how much imagination that scientists need to use to conceptualize what's going on, whether it's what's happening in outer space or what's happening at the microbial level. And one of my concerns in terms of the education system is that children are not having that freedom, sort of free play where they exercise their imagination. I was just interested to hear from you where you think the way you've spoken this evening, you've obviously had to kind of really think and imagine what's going on to conceptualize these very complex processes, whether they're in the body or in the river, and where that your sort of imaginative as well as your ability to kind of reason, as it were, comes from. And is there something in your education in your childhood that really facilitated that?
That's a really, really good point, and I couldn't agree more. I don't think I'm particularly imaginative or creative in any way, but I do agree wholeheartedly that the current education system is too constraining. I do sometimes look back and think, was there something in the way I was taught or brought up that allowed me to do that? I think there were some elements. There were some professors at university even that were very free. We got to do more. They gave us more scope to do our own thing. I'll just give one example. Actually, it was in a practical class. He became my PhD supervisor, perhaps not surprisingly. We were just given a bunch of materials and allowed to design our own experiments. And those practical classes went on for several weeks. And for that to happen now, I can't do that. I can't teach like that because of certain constraints in terms of lab time and, driven by numbers of students and so on. We get to do it to a certain extent when people can come into our labs for research projects. And I do like to offer that scope. So it is important, and some people struggle with it. Some people need more focus, and you try and direction, and you need to strike that balance depending on the person. So I don't know if there was any particular sort of extra scope that I was given other than that. And what I think I'm trying to think about how we can do better for our own students. And I think you're absolutely right. I think giving more space and the facilities for those who need it for hands-on science to do that and do that at a very young age and just encourage exploration. And I wrote a little article. It was going to be a little editorial, but it turned out to be a monster on excursions in science to improve specifically microbiology literacy, but it could have been written for any science really. And it was written during the COVID crisis, or the COVID crisis came on board just as it was about to be published. So it's probably the worst time editorial ever. But that got me thinking about all the different ways in which school children especially can explore their environment. And it doesn't have to be, you know, going far. It can be to the school pond, and what tools we can offer to help teachers to help children, especially to explore in that way. So sorry, I've waffled on a little bit there, but coming back to your main point, I agree wholeheartedly. You do need a good degree of imagination, but to think about an invisible world, you also need to, you know, to sit down and read and understand what's going on in somehow a very focused and constrained way.
Thank you.
I would just, you know, add or maybe disagree in the lack of imagination that you say you have, Terry. I think Lisa started saying that that before perhaps in the interdisciplinary disciplines permeated more, and I think imagination is one of those things that we share. Is not only the role of artists. So that's what I'm saying, that imagination is there in the science, and so we get to share that, and that's common ground, I think, for us.
Yeah, I think one of the, you know, sometimes when scientists are discussing a particular phenomenon, they will actually say, well, we actually haven't been able to imagine this next step, to conceptualize. I've heard this several times on, you know, in discussions, and these are discussions for the public. They're not closed, you know, scientific conferences or so I mean, I agree with what Emilia is saying. It often stuns me, you know. So
yeah, it stuns me too. And you know, you think of theoretical physics, where, you know, those guys and needs huge imagination to think about what's going on at a subatomic level. It blows my mind too.
Yeah.
I think it's in line with what I was saying as well about just how inseparable nature and culture are, right? Or if you think of how inseparable science is from maybe like the social science, there's a very human aspect to the conclusions that scientists will draw. It's based on their history, their geography, their sex, race, etc. Yeah, so that and I think that's what a lot of the artworks in the exhibition talk about, particularly from Escala. And I don't know, I want to gauge what everyone's appetite is right here. We could end it here because we are 10 minutes past, but I am sad that we didn't get to hear you speak a little bit more about the artworks, or any of the artworks that you found interesting, Terry. Maybe if you have like 2 minutes just to say which ones you liked and why. Or
yeah,
if not, don't worry, we can end it here. Yeah.
No, I'm happy just to do that. I won't waffle on too much. So let me. I don't want to get the wrong. Well, I'm going to embarrass Emilio. I'll just briefly mention why I liked the Salto de Tequendama. I'm sorry for my pronunciation. Yeah, let's have a picture. You know, this was incredibly striking. I actually visited the gallery as well this afternoon, so it was brilliant to actually see it in reality.
Great.
And since I've embarrassed you, I'll embarrass myself, because the reason why I got excited by that in the first instant is because I saw the word salto and I thought salt. Of course, salto does not mean salt in Spanish or Portuguese. It's fall, or yeah. So it relates to the waterfall Tequendama, and it's really beautiful and striking. But why I got excited not only was the word salt, I actually thought they looked like layers in a microbial mat, a bit like the one I sent you the image of, but certainly what you sometimes see at the base of these salinas, these sultans. You often see very highly colored greens, golds and black and and read. And yeah, I thought that might have inspired you, but clearly not. But irrespective of that, I think it's a great piece of art. So thanks for that. And the other, sorry.
No, go ahead.
Yeah, no, the other one that really struck me, that I loved, was the El Charquito video. This was relating to the Bogota River. What was fascinating about this, and I really do recommend people to watch this. Yeah, there's one of the fellas. So there were 4 elders who were interviewed who live in El Charquito, a small town that grew up around a hydroelectric development. And it's about the sense of community. It's about how they saw what was going on around them, that's kind of the growth, and then the terrible pollution of the river that followed as a consequence of growth and mining and so on and so forth. And it was just brilliantly done, in the sense that the people chosen were all very different, but they were all supremely honest, working people. And they came to the town to escape violence, for work, and they were all so proud of their achievements, and they all really sensed that how the river was at the heart of the town, and that its demise was and is a major issue. So yeah, I thought that was tremendous, and I wholeheartedly recommend it.
Okay, thank you so much. That was amazing. I wish we had more time to just do that, go through every work and get a microbiological perspective on it. Would be amazing. But yeah, I just want to say thank you to everyone who stuck around. We are a little bit over time, and wanted to apologize as well for the technical difficulties there at the beginning. I think even a year on, we're still all trying to adjust to Zoom and the different dynamics that being online necessarily entails. But thank you to Terry, especially for coming to speak with us and for sharing this hour with us. It was really, really fascinating. And yeah, hopefully we can do this again with maybe a bit more time as well.
Yeah. Thank you all very, very much. I've enjoyed it. Thank you.
Thanks a lot, Terry.
Thanks, Terry. It's fantastic. Thanks, everyone.